So I do apologize that we don't have pictures of the mice. They often remove these for copyright issues. But basically, it was just 2 mice that were there. One mouse was light colored, like white, and the other one was dark colored, like gray, black kind of thing. Hold on, I'm just going to pause the recording. All right. Apologies for the interruption. Okay, so here is the first example for our genetics questions. Before we actually go into it, I just want to show you guys some examples of kind of the difference between these externals. Let me just pause this real quick and pull it up. Right. So I've been going on about No Brain Too Small. It's a really great resource for preparing for external exams. The reason why is they have a lot of things. So mechanics people, can we be quiet? I know you're explaining it, but I'm trying to give some notes. John, shh, inside voice. Okay. There's only like 15 minutes left. And then come back. Please. Give your note. I don't want you in trouble.
That's all right. I'll just tell him.
Okay. All right. So I know a lot of you guys are doing acids and bases as well as genetics, which is a good combo. I just want to show you guys the difference by what I mean when it comes to like nice set categories versus not nice set categories. So when we look at acids and bases, one of the things that I want you guys to kind of keep in mind that is provided by No Brain Too Small is that they collate their questions. So what they do is they go through every single year exam and they pull out the questions that are similar to each other. So that way you guys can study based off of a theme or a topic instead of just trying to study everything at once. A really useful exam strategy is to study things that you are struggling with first, and then the stuff that you are doing really well on to study last. Acids and bases has 3 set questions, and they are always going to be in those 3 categories. So it makes it really easy to study. They have atoms and ion as their first question. The second question is going to be on acids and bases, and then the third question is going to be on rates of reaction. So they're always grouped nicely like that. The tricky thing with genetics is that it is not like that because genetics is a far wider breadth of material. So when we go to look at their collated questions, they have 4 different ones. They have DNA and the role of DNA. They have Punnett squares and pedigrees. They have variation and survival. And then they have a mixture of like older exam questions as well. What they're going to do with the genetics exam is they're going to chop and change and move things around. So there's not going to be a nice set category to study. So the tricky thing with genetics is basically you kind of have to know everything to be able to attempt the exam question because of how they chop and change it. So that's something to keep in mind with genetics. I would personally say that genetics, unfortunately, is the hardest out of the 3 exams when it comes to getting the excellence, and even I would say even the achieved. Genetics has a lot of big concepts and vocabulary. I would say actually the easiest of the 3 exams is probably mechanics if you're good at math. There's not a lot of writing. The trick with genetics to make sure you're going to pass it is knowing your vocabulary, because if you know your vocabulary, you can decode the question. The other thing to keep in mind with genetics is that if you are able to give me the definition, you will be able to get achieved. So worst case scenario, if you don't understand the question, just give me the definition to the words that you see in the question itself. Are we okay so far? All right. So let's look at that first exam question I've given you. So this one's from 2016. It is a bit older, but that's okay. They're all relevant. They haven't changed the standard. So in this case, we are talking about 2 mice. We have a mouse that has dark fur and a mouse that has light fur. And they want you to use this example to talk about how DNA controls appearance. So that relationship between our genetic information and our phenotype, what we physically express. With all of these questions, it's the same basic core concept about DNA, alleles, and genes, with a different example projected onto it. So they might just change the animal, or they might give you a plant instead. The thing to remember for a lot of the excellence points is they want you to tie it back to the question. So tie it back to the example they are using. Make sure you're talking about the mice then. All right? So they want you in your answer to refer to DNA-based sequence, genes, and alleles. I like to underline things because it helps me kind of keep an idea of what I might need to cover. This is an example of definitions. So if you can write down the definition for gene and allele, that can help you get your achieved points. So if you're stuck and you're like, I don't know how to tackle this question, just give me definitions, okay? The other thing to keep in mind is that the genetics exam is far more open-ended. It's not as scaffolded. So they just give you this and they're like, here's all these lines, write an answer, which is quite intimidating. So what I want us to think about first is what is DNA? That would be my first kind of tackling of this question. So we know that DNA is made up of a base. That base is attached to a sugar. And then that sugar is attached to a phosphate. So that's the basic structure that we know for my DNA structure. Are we okay with that so far? All right. So what we have then is a nice double helix. Just trying to think which way I want to do it. Okay. Oof, I'm so bad at drawing squiggly lines. All right. Ooh, that's really bad. I'm sorry. I'm not a good drawer. All right. Feel free to draw things on the exam. It's a really good way to describe what's going on. Don't worry, I am recording it, and I will scan these notes. Just want to make sure because you guys look panicked. All right. So DNA kind of looks like that as our kind of helix. Are we okay with that so far? And if I was to zoom up on one of those little helixes, this guy over here, that is what that is up there. Are we okay so far? All right. So that kind of gives me an idea of what we're talking about when we talk about DNA. The other thing we need to talk about with DNA is what are some of those terminologies that we are referring to. So gene and allele are two good examples of what we're trying to refer to. A gene is a section of DNA. Just watching our time. We might need to finish this tomorrow, which is fine. It's kind of like our roadmap. We're trying to state where on the road of our DNA sequence are we interested in. This gene codes for a trait. So this trait that we are looking at here is going to be fur color. We good? Okay. The next layer of this answer then is the sequence of that DNA. So you guys hopefully have been learning about the 4 base codes. You have A, which binds with, now I gotta remember, T, thank you. And then you have G that binds with C. So those are our bases, which is these guys up here. A, T, G, and C, that is basically like our computer program that tells us how to make life. Now, this section here could have more than one sequence. Say, for example, my initial sequence is A, T, G, C, T, T, A. And if I wanted to do the base pairing for that, it would be T, 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 A, C, G, A, T. All right, so that's an example of one of the sequences of these guys. Sequence number 1. Okay? Now, what can happen is I could have a mutation occur. So a mutation is a change in my base sequence. So for example, oh, I don't want the yellow, I want the pink. For example, let's say there's a mutation on the 4th code pair. Everything else stays the same. So let's say that's A. Let's now change that to a G. So there's a mutation that occurs here. It is now a G. And then the rest of it stays the same. G, C, T, T, A. And then let's write up the pairing for it. So this then is sequence number 2. Are we okay so far? All right. So we have had a mutation. We have a new sequence. These sequences are really important because they then determine the color of the fur. So for example, let's say the original sequence here, this codes for brown fur or dark fur. Did they say dark or brown? They said dark. So let's say dark. We'll use the same terminology. So this one's dark fur. A mutation has occurred in either my egg or my sperm, and now my offspring has light fur. Okay with that so far? Yeah. All right. Last thing I'll just say before the bell goes, these differences in the sequence, those are alleles. All right, we'll finish this tomorrow. Oh, I will take any of the measurements, please hand in. Bio, internal people, hand in. Just leave it on my desk. I'll organize it. I haven't filled it in yet.